Structure and functions of mammalian blood. The mammalian blood consists of a pale yellow colored fluid known as plasma. And in this plasma, there are suspended several types of cells and fragments of cells. The chief function of the blood is to transport oxygen and food materials to the tissues and remove carbon dioxide and other waste materials from the tissues to their sites of excretion. So generally, the following are the functions of mammalian blood. Transport of oxygen from the lungs to the other parts of the body, and also transport of carbon dioxide from the various parts of the body to the lungs. Transport of nutrients from the small intestines to other parts of the body. Transport of waste materials such as urea from the tissues where they are produced to organs where they are excreted. Transport of hormones from endocrine glands to their target organs. Blood is also involved in regulation of body temperature by distributing heat from where the heat is released in organs such as the liver and also skeletal muscles to other parts of the body. Blood also has a defense function against invading pathogens. So, the blood is a specialized tissue, as I mentioned, that is made up of pale yellow fluid known as plasma, and within this plasma there are suspended cells, red blood cells, white blood cells, and cellular fragments known as platelets. We'll start by looking at the plasma. Plasma consists of 90% water. In fact, 90% of the plasma is water, and the remaining 10% is made up of dissolved substances. Now, about 70% of the dissolved substances are plasma proteins, such as albumins, globulins, and fibrinogen. So the remaining 30% comprises of dissolved organic food materials such as sugars, mainly glucose, amino acids, inorganic salts, and also waste materials such as urea. So in terms of volume, the plasma constitutes about 55% of the blood. Then the remaining comprises of, that is 45% is red blood cell, and only about 1% is made up of white blood cells and the platelets. So within the blood, you have the plasma, in which the other blood components are suspended. Plasma serves the following functions of blood. 1. Transport of nutrients from the small intestines to the other parts of the body. These nutrients are in the form of amino acids and sugars that are transported in soluble form. 2. Transport of waste materials from the tissues where they are produced to the organs such as the kidney where they are excreted. 3. Plasma also transports hormones from the glands that secrete them. For example, from the pituitary gland where ADH is produced to the kidneys where the antidiuretic hormone, that is the ADH, functions. So plasma is involved in the transport of hormones from the glands that secrete them to their target organs. And 4, plasma, being mainly water, is involved in the regulation of body temperature by transporting heat from the highly active organs, such as the liver, to the rest of the body. Next, look at the red blood cells. It's the most numerous cells that make up the blood. So, red blood cells, also known as erythrocytes. Red blood cells are very small cells, about 8 micrometers in length. So at the longest part, they're about 8 micrometers in length, and the thickness at the longest part of about 2.5 micrometers. They are sunk at the center. They're sunk at the center, and this gives the red blood cells a biconcave disc shape. So the red blood cells are characteristically biconcave in shape. In a healthy adult human being, there are about 5 million red blood cells per 1 cubic centimeters of blood. So they are numerous. 5 million red blood cells in 1 cubic centimeters of blood. That's a very large number. Now, red blood cells are enclosed by a very thin plasma membrane, and this helps to promote a fast rate of diffusion of gases, especially into and out of the cell. The cells also lack a nucleus. At maturity, they lack a nucleus and many organelles. Instead, the interior is filled with a red pigment known as hemoglobin. This pigment binds and transports oxygen. So this makes the principal function of the red blood cells to be transport of oxygen from the lungs to the other parts of the body. Now, the red blood cells have the following adaptive features that enable them to transport oxygen efficiently. One, the biconcave disc shape, which provides a large surface area for exchange of gases through diffusion. Two, the absence of a nucleus and other organelles, and instead the presence of hemoglobin, which has a high affinity for oxygen. That is, hemoglobin readily combines with oxygen in areas of high oxygen concentration and to form oxyhemoglobin. So in areas of low oxygen concentration, the oxyhemoglobin will break down into hemoglobin and oxygen. Now, as I mentioned, the absence of the nucleus and other internal organelles provide space for more hemoglobin to be filled. That's another adaptive feature. So this lack of a nucleus and other organelles provide space for more hemoglobin to be filled in, and thus increasing the efficiency of oxygen transportation. Even though the absence of the nucleus greatly reduces the lifespan of the red blood cells. They have a very short lifespan of about 120 days. Another adaptive feature is the thin plasma membrane, which allows for rapid diffusion of gases. functions of the red blood cells, as mentioned before, one, the main function is transport of oxygen from the lungs to the tissue. Within the red blood cells, as I mentioned, there is hemoglobin. Hemoglobin combines with oxygen to form oxyhemoglobin. Oxyhemoglobin is bright red in color and is responsible for the bright color of arterial blood. Hemoglobin alone, pure hemoglobin alone, is dark red in color and hence is responsible for the dark color of the venous blood, that is the blood that is found in the veins. So the blood in the vein is dark red because hemoglobin is unbounded. Blood in the arteries is bright red because most of the hemoglobin have been bound to oxygen to form the bright red oxyhemoglobin. So by combining with hemoglobin and forming oxyhemoglobin, the efficiency of transportation of oxygen is greatly enhanced. In fact, 97% of oxygen is transported in the form of oxyhemoglobin and only 3% in solution form. The other function of the red blood cells is the transport of carbon dioxide. Carbon dioxide is formed in the tissues as a result of cellular respiration. Then it diffuses from the tissues into the bloodstream and is transported to the lungs. Now, about 80% of carbon dioxide in the bloodstream enters the red blood cells, where it combines with water to form carbonic acid. So you have carbonic acid that is formed from the combination of carbon dioxide and water. That is a reaction. Now, in this process is catalyzed by an enzyme known as carbonic anhydrase. Carbonic anhydrase. The presence of the carbonic anhydrase greatly speeds up the uploading of carbon dioxide into the red blood cells. Carbonic acid then dissociates into hydrogen carbonate and hydrogen. So you have this dissociation, hydrogen and hydrogen carbonate. The hydrogen ions combine with oxyhemoglobin, forming hemoglobinic acid, thereby facilitating the release of oxygen by oxyhemoglobin, while the hydrogen carbonate ions pass out of the red blood cells and get into the plasma. It is in this form that the carbon dioxide is transported to the lungs. Once in the lungs, where the concentration of carbon dioxide is low, the hydrogen carbonate ions enter the red blood cells, combine with hydrogen ions, forming carbonic acid. Carbonic acid then dissociates into carbon dioxide and water. The carbon dioxide diffuses out of the blood into the lungs and then it is exhaled. So, red blood cells are involved in the transport of carbon dioxide. In fact, about 80% of the carbon dioxide is transported in the form of hydrogen carbonate ions, and this is made possible by the reaction that takes place within the red blood cell, which is catalyzed by carbonic anhydrase. Now, note that about 15% of the carbon dioxide combines with the hemoglobin also to form carbaminohemoglobin. So this is another form in which carbon dioxide is transported in the form of carbaminohemoglobin. Now, in the lungs, the carb-aminohemoglobin also dissociates, release the carbon dioxide, which is then exhaled. Now, next look at the white blood cells. The white blood cells. Now, unlike the red blood cells, white blood cells have a nucleus, but they lack a pigment, they lack hemoglobin. Hence, the cytoplasm is colorless. In terms of numbers, there are much fewer white blood cells than the red blood cells. In fact, for every one white blood cell, there are about 600 red blood cells. However, the number of the white blood cells increases greatly during an infection. White blood cells are formed in the bone marrow of long bones, and like the red blood cells, which are formed in the bone marrow of short bones, like the vertebral column and the ribs. The white blood cells are formed in the marrow of long bones and also in the lymph nodes. Now, there are two types or groups of white blood cells. White blood cells, also known as leukocytes, are grouped into two, namely granulocytes and agranulocytes. Now, granulocytes are so called because they contain within their cytoplasm granules. These granules give them this, appear as dots. So granulocytes are so called because of the presence of granules, protein granules, within their cytoplasm. These granulocytes have amoeboid movement. They are capable of changing their shape to pass through the walls of the capillaries and into infected tissues. In the tissues and the blood, these granulocytes engulf pathogenic microorganisms through the process called phagocytosis. Hence, granulocytes are also known as phagocytes because they do destroy microorganisms and other unwanted materials through phagocytosis. Once ingested, the microorganisms are digested. Now, some white blood cells may die in the course of phagocytosis. So the dead phagocytes, together with the dead microorganisms and damaged tissues, form what is known as pus. There are three types of granulocytes: a neutrophil, a eosinophil, and basophil. That is the neutrophil, eosinophil, and basophil. Granulocytes, on the other hand, lack granules in the cytoplasm, and they tend to have either a rounded nucleus, like in the lymphocyte, or bean-shaped nucleus, like in the monocyte. So the monocyte have bean-shaped nucleus, while the lymphocyte have rounded nucleus. Now, monocytes are produced in the long bone marrow, just like the granulocytes, and they are also phagocytic. That is, they destroy pathogens through phagocytosis. Lymphocytes, on the other hand, are formed in the lymph nodes, and they produce antibodies that protect the body from infection in a number of ways. One, There are those lymphocytes that produce or they act as antitoxins. That is, the antibodies that produce act as antitoxins. These antitoxins produced by the lymphocytes neutralize the toxins or antigens produced by the pathogenic microorganisms. So, some forms of antibodies produced by certain types of lymphocytes are known as antitoxins that neutralize toxic materials from pathogens. In other lymphocytes, the antibodies produced are known as agglutinins. Agglutinins cause clumping together or agglutination of microorganisms. This stops the microorganisms from multiplying and eventually they die. In this way, it makes it easier for the phagocytes to ingest the clumped microorganisms. Still, other lymphocytes produce antibodies that are known as lysins. Lysins destroy microorganisms by digesting their cell membranes or walls, thereby rupturing them. Now, once their walls or membranes are punctured, then the cells are destroyed and microorganisms die. Then, you also have lymphocytes that produce antibodies that are known as opsonins. Opsonins adhere to the outer surface of microorganisms, thus making them easier for the phagocytes to recognize and ingest them. Opsonins are like markers. So the antibodies are produced when microorganisms invade the body, and they attach themselves onto the invading microorganisms, thereby marking them and making it easier for the phagocytes to identify and digest them. So those are the various functions of the different types of leukocytes, because the function of the leukocytes depend on the type. Granulocytes are phagocytic, just like the monocyte also, and then the lymphocytes produce antibodies, different types of antibodies that together or generally act to destroy microorganisms that cause diseases. Now, the third component, actually the fourth component of the blood, are the platelets, also known as thrombocytes. Platelets, also known as the thrombocytes. Platelets are fragments from large cells in the bone marrow. They are in fact cellular fragments. They have a complex structure and arise from the division or the breakdown of the cytoplasm of giant cells in the bone marrow known as megakaryocytes. So megakaryocytes break down to produce these platelets. So these are the platelets. They are regular in shape. Platelets don't have a nucleus, but they have several organelles. Their function is to prevent blood loss after damage of blood vessels through the process of blood clotting. So the platelets do this by forming a plug in the wall of the damaged vessel by producing a substance known as thromboplastin. Thromboplastin then initiates the blood clotting process. Now, the blood clotting process is very important as it prevents loss of blood and also prevents the entry of pathogens. So we now look at the blood clotting process. that is triggered off when a blood vessel is damaged. The process of clotting involves a series of complex reactions where soluble blood protein is converted into a mass of tangled threads known as fibrin that stops the bleeding. Now, when the blood vessels are injured, for example here, when this blood vessel is punctured by the pin, the walls of the vessel is damaged. So this damaged wall produces a rough surface that stimulates the platelets to release an enzyme known as thromboplastin. So here we have the damaged blood vessel. This rough surface, as a result of the damage, will stimulate the platelets to release thromboplastin. Or even exposure of blood to air stimulates the platelets to release the enzyme thromboplastin. Now, thromboplastin achieves two things. One, it neutralizes the anti-clotting agent known as heparin. Heparin is a protein that is present in the blood and prevents clotting, blood clotting. So under normal circumstances, the blood will not clot, and the clotting process is prevented by the presence of heparin. So thromboplastin neutralizes heparin. Then secondly, the thromboplastin stimulates or activates the inactive prothrombin, it's another protein, to the active form thrombin. Prothrombin is synthesized in the liver and requires the presence of vitamin K. So prothrombin is one of the plasma proteins, ordinarily to not be activated unless there is a presence of thromboplastin. Under normal circumstances, it is the heparin that prevents the activation of the prothrombin. So the release of thromboplastin by the platelets first neutralizes the heparin, and secondly, activates the prothrombin into thrombin. This activation process also requires the presence of calcium ions. Prothrombin is made in the liver, and it requires vitamin K input. Then it is released into the bloodstream. On exposure, platelets release thromboplastin. Thromboplastin then activates prothrombin to thrombin. And this conversion requires the presence of calcium ions. The active thrombin then stimulates the conversion of the soluble protein fibrinogen into the insoluble fibrin. The fibrin forms a meshwork of fibers. On the cut surface that trap the red blood cells to form a clot. So this fibrin forms strands that are insoluble on the surface of the damaged blood vessel. So this traps the red blood cells, and the trapped red blood cells therefore will prevent further loss of not only blood cells but even the body fluids. So as the clot dries up, it forms a scab. As it dries up, it forms a scab that stops the bleeding and protects the damaged tissues from infection. So this process of blood clotting is summarized in this diagram here. First, there's the damage to the blood vessel. That damage to the vessel stimulates the platelets release thromboplastin. Thromboplastin then neutralizes heparin and also activates prothrombin into thrombin. Thrombin then activates the conversion of the soluble fibrinogen into the insoluble fibrin. The insoluble fibrin fibers then form a meshwork above the cut surface here to stop further bleeding and also prevent infection by pathogens. Thus, blood clotting reduces loss of body fluids. Now, excessive loss of blood, especially more than 2 liters, can lead to severe anemia unless it is corrected by blood transfusion. Now, it is important to note that in humans, there are some individuals that lack the clotting factors, the factors that are necessary for the conversion of prothrombin to thrombin or fibrinogen to fibrin. This condition is known as hemophilia. When any of the factors along the step are absent, so that the process cannot be completed, then we have a condition known as hemophilia. Hemophilia may arise due to genetic disorder or can be due to lack of vitamin K. Individuals that are afflicted with hemophilia may bleed excessively from minor cuts, which can be fatal. Now, injections of vitamins containing vitamins K may be given to prevent excessive bleeding if the cause of hemophilia is lack of vitamin K.